Hello, everyone, and welcome to today's live keynote presentation, Treating with Cannabis, presented by Jeff Hergenrather, MD, consultation physician, president of the Society of Cannabis Clinicians, vice president of the American Academy of Cannabinoid Medicine, Northern California. I am Christina Mahalik of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, please click on the Help Desk button located in the promotional board at the bottom center of your screen, or use the Ask a Question box to let us know you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Please join me in welcoming Dr. Hergenrather. I will now turn the presentation over to him.
Hello, thank you for the introduction. I'm a practicing physician in Northern California. I've been doing cannabis consultations for about 20 years. My experience with cannabis really dates back about 40 years, but since California changed our laws in 1996, I began to use cannabis as a part of my practice, and a couple years later opened an office specifically doing cannabis consultations and have done that since then. I am in a solo private practice, and I have no financial relationships to disclose. So though I may mention types of products, I do not have any financial relationships with these. So I think one place to start would be to simply say that things have changed in the United States in the last few years. At this time in polling the public, nearly 70% of Americans live in states that have reformed their marijuana laws by allowing medical marijuana. And polls are showing that 60% of Americans should support making marijuana use legal altogether. And about 90% support allowing medical marijuana. Things changed a lot in 2013 when CNN chief neurosurgeon and correspondent to the network came on and told the story about a young child in Colorado who has a seizure disorder. And he realized after looking into this case that this girl was benefiting enormously from the use of a high CBD form of cannabis. After the presentation, he went on to say that he had been terribly and systematically misled for nearly 70 years in the United States, or we have been terribly and systematically misled for nearly 70 years in the United States, and he apologizes for his own role in that. So he really turned his opinion around in seeing the use of cannabis in a childhood seizure disorder. This seemed to correspond to the public feeling a lot more comfortable asking the question of what about marijuana? What about cannabis as a useful medicine, not only for seizure disorders, but for all kinds of disorders? The public has changed the laws now in many states. We have 23 states with medical marijuana and 6 more that have medical and or adult use of medical marijuana. So 29 states now have approved the use of cannabis on a regular basis. There are some limited access states that are allowing the high CBD strains of cannabis, the ones with very reduced psychoactivity, in another 16 states. So there are only 5 states now that have no medical use and even they are talking about changing their rules. Yet we still have no change at the federal government, so we're still working in a climate of prohibition. The only state that changed the laws by way of their legislature is Vermont, which is interesting. So what I'm saying is it's the public that has changed the laws, and it's the opinions in the public that have really come around a long way because people realize and find that cannabis is so useful for so many things, or they hear about it in their friends and relatives. So this is a map. It's only missing West Virginia as being shown here as a no medical marijuana state. In fact, it does have a medical marijuana law now, and that was just about a year ago when they changed their law. So you can see, except for the gray states, there's cannabis use coming more or less all over the country. This whole issue about cannabis as a medicine comes from our knowledge of the endocannabinoid system. This is a system that's built into the body that brings us into balance. We didn't know about it until really the last 20 or 30 years, when a cannabis-like molecule was radioactively labeled and injected into the body, and it went to very specific locations. This gave us some information about where this system could be found, and it shows up in our neurologic system, in the brain and in the nerves, and it also shows up in the immune system. It is made up of receptors that I would describe just about like this, and ligands or molecules that activate these receptors, that fit into these receptors on the surfaces of cells in the immune system and in the nervous system. So what are the roles of this natural system? It helps us to eat and to sleep and to relax, both physically and mentally, to forget in a good kind of a way. It extinguishes bad memories. It helps us to get fear and pain off of our minds. It is very important in the way we learn and forget. Protection, it protects in many ways. So the immune system is basically scouting around in the body, looking for problems and programming cell death of abnormal cells. So it has a role in protecting cells, protecting nerves, and killing cancer cells as well. So what I'm showing you here is that the endocannabinoid system is seen from hydra to sea squirts, fish, really all throughout the animal kingdom, perhaps with the exception of insects. We don't really have much evidence that the insects have an endocannabinoid system, but all other animals in the animal kingdom do. We see it in the genes, and this has been preserved for millions and millions of years of development. As we've evolved along, this particular system is key to our survival, and it can be activated by cannabis. So going back to the radioactively labeled cannabinoid, you can see the areas that are highlighted in reds and orange and yellow are the areas most concentrated in the cannabinoid receptors in the body or in the brain. So the middle row of column is the MRI scan, and the right-hand column is a blend of the receptors with the MRI images. So you can see that the brain is rich in these receptors, and they have very specific roles depending on where they are in the brain. In the rest of the body, this was injecting a cannabinoid into the body with a radioactive tracer, and we see it at the time of injection showing up immediately in the liver, the bright red spot on the left side of this image, and in the skeleton it's showing up. So as the minutes go along, it goes from 0 up to 102 minutes. You can see that these receptors, again, are lighting up wherever they are in the body. They are primarily in the immune system in the body, looking again for trouble, if you will. So on the CB1 side, the ones that are in the central nervous system, we know that they're not only in the brain, in the concentrations that I was indicating, but they also go in the nerves throughout the body into all of these different locations. And on the CB2 receptors, a slightly different receptor, but closely, a very close approximation of the CB1 receptor. It's in these immune cells, the monocytes, the macrophages, the T cells and B cells, liver, spleen, and tonsils, all involved in immune responses. They also cluster in the brain and in the gastrointestinal tract. So what are the roles of these cannabinoids when they're activated? When the natural cannabinoids, the ones that are built in, that are very similar to the plant cannabis cannabinoids, they activate these receptors and provide these various pharmacologic effects. This is not an entirely complete list, but it gives you a pretty good idea of what all they can do. So by activating the receptor, we have pain relief, antispastic effects, anti-anorectic, which means they help the appetite. They reduce vomiting. They're neuroprotectants. They have anti-cancer roles, antioxidant, antibacterial, antifungal, antiparasitic roles. They're anti-inflammatory, said to be as potent milligram per milligram as steroids. There's immunosuppressive roles that are very useful, and I see great responses in people with transplants of their organs using cannabis and not needing to use as much of their immune modulating drugs. There are a number of dermatologic effects that are very important. They're bronchodilating as opposed to bronchoconstricting. So even though people might use them as smoke or vapor, they're actually opening up the airways rather than constricting them. That's not to say it's not an irritant to the airway, but because it is. It's an anti-glaucoma agent, anti-diabetic, bone stimulating, so it strengthens our bones with the cannabinoid activation. They're antipsychotic, anxiolytic, anti-anxiety medicines. They're antidepressants. They're vasorelaxants. They allow blood vessels to relax and carry more blood. They're anti-ischemic. In the first studies that were done about cannabis decades ago, we learned that they can protect from the size of a stroke. They're anticonvulsants. And then they have numerous roles in the gastrointestinal tract. They slow the motility, they reduce the secretions, they reduce stomach acid, and they reduce reflux. They also help induce sleep. They shorten the duration of time getting to sleep at night. So the natural cannabinoids are represented by these two molecules. There are actually more of them, but these are the predominant two that are seen throughout the body, anandamide on the top and 2-HE on the left. The next slide shows the plant cannabinoids, or at least three of what are now counted to be between 100 and 140 different cannabinoids in the cannabis plant. Depends on where the strain is grown and what the genes are like in that particular area. You'll see delta-9 THC on the upper corner and CBD next to it on the right-hand side. What I'm showing this to indicate is that there's a very subtle difference here between these two molecules, right in this point of the ring, where a hydrogen atom comes from one point of a methyl group over to the oxygen, saturates that oxygen, opens the ring, and thereby changes the shape of the molecule ever so slightly. It's all the same atoms. Its atomic weight is all the same as well. It's just a slightly different configuration that does not fit in the cannabinoid receptor quite the same way. So yes, it can get into the receptor, but the CBD doesn't activate the receptor. Its work is somewhere else. The THC actually gets into the receptor and, very much like the natural cannabinoids, activates those receptors to provide all of those different effects. The bottom is just another example to show you something about the structure-activity relationship. The THCV is another plant cannabinoid, and you'll see right here at the bottom, the chain is a little bit shorter. It's 3-carbon chain as opposed to a 5-carbon chain in THC. And I think that what we know about this is that THCV gets into the cannabinoid receptor, but it doesn't activate it unless it's in very high concentration. So by having a shorter tail, if you will, or side chain, it does not have the same effect on the receptor. Basically, it acts like a mild blocker of the receptor. So this is a look at what the receptor might be like. On the left-hand screen, we see dots which represent amino acids. So in the case of the CB1 receptor, there are 472 amino acids that are a single continuous chain, and when it clumps, it makes a socket, if you will, that moves from the inside of the cell up into the membrane, and there it's ready for business. The CB2 receptor is the green dots, and it too has forms a receptor very much like the CB1 receptor. It's a little shorter, but it does basically the same thing in that mobile group of cells that are around the body. It's known from the basic research that if you change just one of these amino acids in the chain, you can alter the function of the receptor. It does not work as well. So the structure of the receptor itself, in very subtle degree, works better or works less well, depending on really the genetics of the receptor genes. So this has led us to understand that there are, in fact, endocannabinoid deficiency diseases. So we can see this variability in these endocannabinoid receptor genes. They're called SNPs, or single nucleotide polymorphisms. And these polymorphisms do affect the functionality of the endocannabinoid system. For some people, this endocannabinoid system just doesn't work as well as it does in another person's endocannabinoid receptor. So this is a couple pages listing some of the conditions that are now implicated as cannabinoid receptor polymorphisms, starting from schizophrenia and migraine, multiple sclerosis and other degenerative diseases, irritable bowel syndrome, failure to thrive. The non-organic failure to thrive is also described as a endocannabinoid deficiency. Anorexia, chronic motion sickness, fibromyalgia, and a variety of menstrual symptoms, which might include severe pain as well as endometriosis, may be endocannabinoid receptor abnormalities. Seizure disorders, even happiness has been recognized as a condition that has a similar polymorphism of the cannabinoid receptor. Depression and other mood disorders, alcohol dependence, obesity, ADD, ADHD, PTSD. I haven't read the papers on this list at the bottom here, but lipid profiles are different in patients with different cannabinoid receptors. Mediterranean hypocaloric diet is different. The risk of cyclical vomiting syndrome may be genetic. And marijuana demand, I'm not quite sure what that refers to. I'll have to read that paper. But let's look at the way that the cannabinoid receptors work in the nervous system. In this case, this is showing a synapse between two nerve cells. One is the presynaptic cell bringing the signal into the synapse, and then the neurotransmitter in this case is glutamate. Now, whether it's glutamate or adrenaline or noradrenaline or dopamine or serotonin or acetylcholine or GABA, they all are the same in terms of the role of the cannabinoid system. The cannabinoid receptors may be in the presynaptic cells, and the cannabinoids are generated on the postsynaptic cell. So as we go through this, we're increasing the glutamate in the receptor, is beginning to cross the cleft and activate the glutamate receptor, and then transmit the nerve impulse from the presynaptic cell to the postsynaptic cell. When this begins to happen, at least in a strong degree, the cannabinoids represented by these shapes are generated. They cross the cleft in the opposite direction to find the cannabinoid receptor. And when the cannabinoid receptor is activated, it blocks the production and the release of the glutamate. So it's a modulator for the way that the nervous system is running. It prevents excitotoxicity from exceeding the safe limits of what the nerves can handle. You can burn up the nerves by overactivating them. So the cannabinoids are there to protect the nerves. Whether it's an excitatory neurotransmitter, as is the case in glutamate, or an inhibitory neurotransmitter, as in GABA, the cannabinoids are there just the same, providing this retroactive retroactive messaging system to let the presynaptic cell know it should back off and stop producing such a great amount of neurotransmitter. In the immune system, it's not so easy to demonstrate it with a graph, but we know some basic things. We know, for example, that there's abundant evidence that the cannabinoids modulate the immune responses. Whether you react or not is a cannabinoid function. Research in South Carolina has shown that THC as a single molecule attenuates the host versus graft reaction, possibly having a role in transplant rejection is what they surmised. And in this bottom study, this was done in Mississippi, and they infected monkeys with the HIV-like virus, the simian immunodeficiency virus. And what they found in those monkeys that got a daily injection of delta-9 THC, basically the main active ingredient in cannabis, is that it ameliorated disease progression, attenuated viral load and tissue inflammation, significantly reducing the morbidity and the mortality of the infected monkeys. So we don't exactly know how this occurs, but we do know that there's a protective role with this particular infection. There are both synthetic and phytocannabinoid medications. This dronabinol has been on the market for about 20 years. It's in 3 strengths. It's not particularly liked by people who use whole plant cannabis because the plant feels a lot better than the individual molecule of THC, or in this case, a synthetic THC. Nabilone is another synthetic THC, and it too is on the market. I haven't seen it used before. Nabiximols are plant cannabinoids, but they're not available in the United States. They are available in 30 other nations as medicines. But we have allowed cannabidiol in a plant extract called Epidiolex, and it is available through centers that specialize in seizure disorders, where we have been able to provide this to children in particular with seizure disorders that really cannot be managed with conventional medications. It is 99% CBD. It's 1% non-THC cannabinoids. I'd like to make a point of the fact that once this plant produced this high CBD product, it went back to the laboratory where they removed all of the additional THC that was there, because there usually is about at least 5% THC in these high CBD plants. They removed it basically for political purposes. They want to see that this is not going to contain any THC, which continues to be vilified as a dangerous drug. When I'm treating kids with seizure disorders, I often will start with THC and CBD, or if I don't get the results I'm looking for with the high CBD products, I'm going to add THC, because THC is a known anticonvulsant and works very well. If you add that to the CBD, you get a stronger anticonvulsant medicine. This is basically an argument for why the whole plant works better than the individual molecules. And rather than reading the whole thing, I want to bring your attention to the non-THC components of the plant. There are a lot of components there, not only these other minor cannabinoids, there are also terpenes, and they're molecules that have some relationship to cannabinoids. There are some terpenes that actually get into the cannabinoid receptor and can activate it, such as beta-caryophyllene gets into the CB2 receptor and can activate that receptor. So there are molecules in the plant, in addition to the THC and the CBD that we commonly talk about, that have numerous therapeutic properties in their own right. So they have, for example, neuroprotective roles, bone stimulating roles, antipsychotic, antiepileptic, antibacterial, antidiabetic, vasorelaxant, anxiolytic, antispasmodic, and analgesic effects. There is a host of pharmacologic effects, so that when the whole plant is used, you get a much stronger effect than you do when you're just using single molecule products. And when given the choice, people uniformly will choose the whole plant because it feels better and it seems to work better than the individual molecule. The federal government knows all about this. For five years ago, coming out of the NIH, these authors stated in their abstract to their paper that modulating endocannabinoid activity, which is what we're doing when we're using cannabis, may have therapeutic potential in almost all diseases affecting humans. Strong words. And then the rest of the laundry list of conditions that they cite, including obesity, metabolic syndrome, diabetes and diabetic complications, neurodegenerative, inflammatory, cardiovascular, liver, gastrointestinal, skin diseases, pain, psychiatric disorders, cachexia, which is a wasting syndrome, cancer, chemotherapy-induced nausea and vomiting, and many others. You know, I would be adding to that autoimmune diseases, insomnia, and other conditions where I really see in my practice that cannabinoids are so beneficial in modulating the endocannabinoid system and helping with those problems as well. This is the federal patent on cannabinoids. It's held by the Department of Health and Human Services. It was applied for in 1999 and granted in 2003. So you can't make a cannabinoid as a medicine unless you're given permission by Health and Human Services and the federal government. There are natural ways to upregulate the endocannabinoid system, such as exercise and massage and manipulation and acupuncture. These will briefly upregulate your natural cannabinoids. And also the metabolic breakdown products of acetaminophen and some of the NSAIDs are recognized as upregulating your natural cannabinoids by altering the metabolism of the natural of the cannabinoids. You upregulate them. So it has a beneficial effect that may, at least in part be, these medicines may in part be from their role in affecting the endocannabinoid system. There are various pathologic conditions that upregulate the endocannabinoid system. Even obesity upregulates the endocannabinoid system. These natural endocannabinoids are activated and synthesized on demand as needed in the body. Then they're rapidly metabolized right there at the site of the action. We do see some of them floating in the bloodstream, but by and large, they're active right at the point where they're needed. The plant cannabinoids, however, are broken down exclusively in the liver in what's called the cytochrome P450 metabolic pathways. And we do have genetic differences in those pathways as well. So one person is going to inactivate cannabis from the cannabis plant in a few hours, and another person is going to take many hours. We'll talk more about this in a minute. The one thing that I think is particularly important here is that there are no significant drug-drug interactions between cannabis and conventional pharmaceuticals, with a rare exception in a drug called clobazam, which is metabolized by the same metabolic pathway as the CBD is. But you've got to have a high dose of CBD for this to be a problem. And so if people are on this particular anticonvulsant, or really I would suggest other anticonvulsants, and you're using a high amount of CBD, you should be looking at your blood levels of those anticonvulsants to be sure you're getting them in the right range. Otherwise, besides anticonvulsants, there's no significant drug-drug interaction between cannabis and any of the pharmaceuticals that are on the market. We can expect a wide range in blood levels depending on a person's receptors and their metabolic pathways. So one dose is not the same from one person to the next. This really shows up when treating patients in a nursing facility for Alzheimer's dementia, where I see a person might get a great effect with 5 milligrams of THC usually in a food like a chocolate or a little snack. And another person might take as much as 30 milligrams or even 40 milligrams to get the effect that you're desiring. So our cannabinoid systems don't work the same, and we really have to adjust the amount of medicine that we use in order to get the desired effect. This is a graph showing the metabolism of smoked cannabis, in particular the THC molecule, as the dotted line. The first pass metabolite, the one that is broken down at first in the liver, goes to this 11-hydroxy-THC. So you see here, after about 5 minutes after smoking cannabis or vaporizing cannabis, you have a peak blood level that rapidly drops down again over this first hour. So at an hour, you can see that probably close to 10% is all that remains from the inhaled dose of medicine. The 11-hydroxy is the one that is in this dashed line, and it's produced by the liver and does end up in the bloodstream. The solid line are the inactive metabolites, abbreviated as this carboxy-THC molecule on the straight line. So as the THC disappears and the 11-hydroxy-THC disappears, we get this solid line of the inactive metabolites that last for days. This is really only useful for law enforcement because these are inactive molecules. They do nothing for us pharmacologically. So just to review, you get the most THC is eliminated over the first hour, but the duration of action is over 2 to 3 hours. Now, looking at the metabolism of ingested cannabis, we have quite a different curve. Now you see that the inactive metabolite shows up first and peaks rapidly over a course of about 2 hours. The THC is the lowest of these curves, and the 11-hydroxy is the greater one. So something about near 80% of the active ingredients is actually this 11-hydroxy-THC. It's a psychoactive molecule, and the THC too. So for the first couple hours, you peak on the effect of ingested cannabis, and then it runs out 6, 8, 10 hours, again, depending on your metabolism. So someone with a rapid metabolism may burn it all up in 5 hours, and another person may take close to 12 hours before they really break down the cannabinoids to their inactive forms. This too has to play into the way that we're teaching people how to use cannabis as medicine for obvious reasons. So just to review this, the ingested cannabis, the THC is eliminated over about 5 to 8 hours, and the duration of action, I would say, is more like 5 to 10 hours. There are a bunch of other therapeutic considerations that are important to consider when talking about cannabis. One of them is that cannabis is described by naturopaths as an adaptogen. So cannabis has a unique ability to give us what we need. That is to say that if you were to use cannabis at bedtime and go to sleep after using it and find that your sleep is more prolonged and is more rapidly, you more rapidly get to sleep. So basically the quality and the duration of sleep is markedly improved. You would also find that using the same cannabis in the daytime is not going to put you to sleep. Your body basically does what you need, and the cannabinoids are there in that role. The method of administration is very important. We have topical uses of cannabis that can be used for skin lesions of all kinds and skin diseases of various kinds, beyond really the scope of this presentation. We speak of vapor and smoke on one hand and ingested products on the other hand. So two major areas where people use cannabis. Also, people are making suppositories out of cannabis. And we don't really know enough about this to advise it in most cases, but there are situations where a person might choose to use a suppository where other methods just aren't practical or effective. Neutral versus acid cannabinoids. This brings up to our point something that I'll touch on as we go forward in the next few slides. But suffice to say that the whole plant, green and growing in the yard, the cannabinoids are in an acid form. And it's not until they're heated that we rapidly decarboxylate these acid forms into the neutral forms of these molecules. There's also the topic of cannabinoid synergism. And I've somewhat alluded to this earlier in saying that different cannabinoids work together better than they do alone. Single plant versus single molecule CBD versus whole plant CBD. these are quite different. And I don't believe I've added a graph, so I'll just speak of that momentarily here. If you take a single molecule CBD product, which you might end up with if you're sourcing it from offshore, we don't really know what's in those unless you take them to the laboratory. Single molecule CBD has a biphasic response curve. That is to say, it may reduce pain and inflammation very nicely up to a point, but as you go up higher on the dose, that will disappear again. So it's biphasic, meaning it goes one way and then it goes another. So we choose to use CBD-rich medicines that are grown as whole plant medicines for this CBD-rich products. And those don't exhibit that biphasic response. You can take a bigger dose and you get a bigger effect. We do want to be able to select cultivars and chemovars as we go into this new era of more and more adult use of marijuana and marijuana for medical use. What we have come to realize is we just have a lot of different chemicals in these products that don't necessarily tell us what we're getting if we're just looking at THC and CBD. There's some studies coming out of Israel that are very telling in treating autistic kids. And that is to say that a very high CBD product, about 20-to-1 CBD to THC, was working great for treating autistic kids. And then suddenly the caregiver said, what'd you do? It's not working. The kids were anxious, they were aggressive. It just seemed like they were back to square zero. And what they found was that even though the THC and the CBD were basically identical in these two products, there were many, many other constituents that were quite different. And, you know, I don't even know the names of these cannabinoids, but in one it was very high and in another it was very low, or vice versa. So there are subtleties of these cannabis cultivars that we don't understand yet. There's too many variables to oversimplify. Our Supreme Court, in a decision about 15 years ago, said that cannabis is a fungible crop. I didn't know the term. Fungible means it's interchangeable with any other cannabis, like a bushel of corn or a bushel of wheat. It's no different from one location to the other. But in cannabis, it is. It is not a fungible crop, and it can't be necessarily substituted between one cannabis strain and another. So we're learning a great deal, but we have a long way to go in understanding what might be best for which conditions. If somebody says that they know the best strain for any particular condition, they're guessing, or there's an anecdote to say that it seemed to work in one person. But as far as really understanding the chemical nature and what's causing one strain to work and one to not work as well, we don't know the answers to those questions. There's another topic here, tolerance and autoregulation, that's very important. And it really takes me back to that image of the receptor in the cell membrane. What we know is that there's a population of these receptors in the cells of the brain, in these brain neurons. And that as cannabis is used on a regular basis, what happens is that some of these receptors will come out of the cell membrane where they're there to receive the cannabinoids, and they'll come back inside the cell inactive. They're ready to be used again if you're not using cannabis, but the body will auto-regulate the population of our cannabinoid receptors. And when this occurs, we don't feel as high under the influence of cannabis. So a chronic cannabis user often will tell you, I'm not really getting high, I'm just feeling relaxed, I'm reducing my pain, I'm doing the things that they might be using it for. So it's really a matter of changing the population of these receptors to where you don't feel high any longer. And this was really well demonstrated by a patient of mine who went home with intentions to work over a course of a month up to a very high dose to see if it would work on a difficult cancer. And when he went home, he just decided, I'm all in. And he took the whole large dose on the first dose. He was wiped. He was on the couch. Now, he had used it in his youth, so he wasn't frightened, but he had to just sleep it off. And a day later, he took a gram of concentrated cannabis oil again. And the third day he did the same. And each day thereafter, he did the same, taking this very large dose, all the while being basically a lump on the couch, really totally incapacitated. But with this autoregulation and tolerance developing, it took about 5 to 7 days, and he was up and around and acting normally again. So even though he could still take the gram of cannabis oil every day, he was not feeling particularly stoned after he had this tolerance developed. So methods of administration. I think this is very useful to understand. Smoke and vapor have a bioavailability ranging from 2 to 56% in the studies done by Hustis back in 2007. In another study, it was in a range of 10 to 25%. But again, a vast range of how much ends up actually in the bloodstream, bioavailable. So you can imagine that if somebody is sort of casually puffing on a cannabis joint and they don't really want to get high, they may just blow it out very readily and not absorb much, not get much into the lungs. On the other hand, somebody that's drawing it in slowly and deeply and holding it in the lungs may absorb more than half of the available cannabinoids that are in the plant matter. So you can get a much higher degree of bioavailability just by the method that you use. This method of smoking and vaporizing gets these molecules into the bloodstream by way of the lungs, and not going by the digestive tract through the liver first and then into the bloodstream. So we say that this avoids first pass metabolism. In oral forms, we say that the bioavailability is 10 to 20%. This is probably pretty accurate, which is to say any dose, if you're measuring all the milligrams of cannabinoids, the amount that actually get into the bloodstream before the liver breaks them down, as I was showing you in that carboxy THC solid line on the graphs, the bioavailability is really quite reduced to about 10 or 20%. We can't really do anything about this. I would say in general, don't eat cannabis products on a full stomach or a big meal. At the same time, it may facilitate absorption to have a small fatty meal to get better absorption. Now, in Hustis's work, she says that bioavailability of rectal or vaginal suppositories of cannabis are somewhere near twice the oral route of absorption with a lower first pass metabolism. What this means is that when a suppository is in the rectal tissue, it's in a watershed zone between the vessels that go back up to the liver first and those that go around into the vena cava and back directly to the heart. So we're right at a watershed in drainage area in the body. And so we do avoid some of this first pass metabolism by the liver when using it in a suppository. The thing that is really not shown here is that these studies on bioavailability of suppositories were done 20 years ago by Mahmoud El-Soli down at the Mississippi farm. Well, I'm not sure if it was there that he did the research, where cannabis is grown for the federal government. But what he was discovering was THC as a single molecule is not absorbed, and that we don't really have studies on looking at cannabis as a plant oil or a whole plant extract. So what he did was devise the best molecule to get it absorbed, and it happens to be one called a hemisuccinate ester of THC. So you're binding THC to a hemisuccinate molecule that is readily absorbed. And it would make a great medicine, but none of these are being produced for commercial use. And I mentioned before, we have topical and transdermal use. Both are very different. One is not getting through the skin and just treating skin conditions topically. The other is designed with solvents to get through the skin and be available as a systemic medication. This is going back to that point I was making earlier about the whole plant. The cannabinoids are in the acid form, and it's withdrawing and heating that they're decarboxylated. This is what's happening. THCA has this molecule. It's called a carboxyl moiety right here stuck to the THC. As you heat it, it comes off as CO2, and you end up with THC. Prolonged drying and storage of cannabis, you'll get a lot of decarboxylation occurring, so that the advantage in THCA is that when used when it's green and fresh is not psychoactive. It's a different molecule. It has different properties. We don't really know that much about it yet. THC, we know much more about. It is psychoactive. So people are using products that are designed with acid forms because they aren't psychoactive. I spoke of synergism earlier. Dr. Costa's work in Italy many years ago showed that rats show a great response to THC and to CBD, but if they're both used together, it's a stronger medicine still. So you get, again, a better effect from a combination of molecules than you do as a single molecule. And McAllister and Marcoux did a paper in San Francisco in 2010 looking at glioblastoma brain tumor cells. And this is one of the graphs that they showed in their paper. What they've done is they've taken brain tumor cells called a glioblastoma, and they've taken a small concentration of THC and added it to a tube of tumor cells. And here, this 100% is just nutrient. The second column is adding that much THC, and it knocks the viability of the cells this much. The CBD is added at this small proportion or a concentration, and it knocks the number of brain tumor cells down this much. And when you put the two together, here's the viability of the cells. So my point is simply, again, THC and CBD work much better together than they do alone. We don't really have a particular ratio that would be best for many clinical trials. This would have to be done as a different experiment. Coming to the end of my talk before questions, I just wanted to show one particular case, and this was a 16-month-old child that was brought to me by his parents when he was diagnosed with a large brain tumor. This is called an optic tract glioma. It's the white mass that we see that was found in August of 2011, right there. It's such a large tumor that surgeons said, "I'm sorry, we can't touch that. It's just simply too big and too deeply seated in the brain." They talked to the radiation docs, and they said, "We have a treatment for you." They talked to the chemotherapy docs, and they said too that we have a treatment for you, but both admitted that they weren't going to necessarily kill the tumor, and they couldn't really say that it wouldn't cause harm to his developing brain. So they found me, and we started to use this concentrated cannabis oil that I spoke of a minute ago with this other cancer patient, squirting a bit of oil onto his pacifier twice a day. This was a solo active treatment. There was no other radiation, chemotherapy, and other than just a great diet and largely vegan diet, that was the only thing that they did. So you'll see 3 months after the initial presentation, his MRI showed a faint image of the tumor. It was just melting away, if you will. And then after 2 more months, it was virtually gone. He has continued to go along year after year up to the present time with no recurrence, while still using a maintenance dose of cannabis. This kiddo is bright as can be, and he's really fun to sit in the office and talk with. When he was still, there's a bit more of a story here. One of the parts of the story is the doctors don't know the truth of what the patients did. They see these images and they call him a miracle baby because they don't know that cannabis was used. The family is frightened of the possibility that Child Protective Services will come out and take their child away. So no one really knows who this baby is. We do see that when this kiddo entered school last year as a first grader, he was taken for IQ testing, and his IQ comes back at 135. So everybody's quite delighted with the fact that he's thriving and doing so well and not showing any sign of the tumor after all of these years. Again, it's just a maintenance dose of cannabis that we're using, and in general, I would recommend that for any cancer that we would be treating. So that's the gist of the presentation up till this point. I thank you for your attention, and I look forward to your questions.
Thank you, Dr. Hergenrather, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your question into the box that appears on your screen and click on the Send button. We'll answer as many questions as we have time for at the end of the presentation. So let's get started with our first question. One moment. Okay, the first question asks, what about damage to the developing brain in pregnancy, nursing mothers, and adolescents?
Well, that's an excellent question, and a question that the National Institute on Drug Abuse is really charged with looking for harm. So there has been a lot of funding going into looking for harm in the developing brain. But really, our first knowledge of this comes from studies that, a study that was done by a Dr. Melanie Dreher when she went to Jamaica back in the 80s and 90s. She collected 30 pregnant women that were heavy cannabis users and 30 control women. And what she found was that the labor, delivery, and the first neonatal month of life was marked by very healthy looking kids coming from the moms that used cannabis. And in fact, they seemed to be more alert and developing even better than the babies that came from the moms that weren't using cannabis. Her initial studies were published in Pediatrics magazine. When she went back years later to see how the kids were doing, the kids from the moms that were pot smokers were still doing better. The Pediatrics Journal was unwilling to publish her findings. They basically said that it may be an Earth Mother phenomenon where perhaps those 30 moms that were pot smokers were just better mothers. But looking for harm in the developing brain has been an area of study for quite a long time now. One of the studies came out of Dunedin in southern island of New Zealand. The study was produced in 2012, and it showed a positive association between adolescent onset of cannabis use and what they said was a decline in the IQ. From this study, Dr. Nora Volkov, our NIDA director, surmised that there is a neurotoxic effect of cannabis on the developing brain that permanently lowers IQ. Well, she says that frequently. I've heard her say it on occasion. But within a year, there was a by Rickelberg, which was talking about a review of this same very study at the University of Oxford. They looked at the same data and said that these conclusions are premature in light of likely confounding from socioeconomic status. And another researcher, Meyer, also suggested that the data, the conclusions from this data by the original authors overestimated and even could be zero as far as a cannabis effect on the IQ of the patients studied. Then brain structure has been studied rather extensively, intensively in the last few years. In 2008, down in Australia, UCell looked at the developing brain, and he suggested from MRI scans that young people, heavy daily cannabis users, he said that the cannabis exerted harmful effects on the brain tissue and mental health. So what he's looking at is the gray matter organs of the brain and saying that there's evidence from their study that there was a difference in these in the gray matter between the cannabis smokers and the non-cannabis smoker controls. But what he didn't really do was check to see whether this proposed gray matter change in the brain was really showing any harm or mental illness. That was not really being, it was being asserted, but it wasn't being really tested for. Following this now, NIDA, the National Institute of Drug Abuse, has gone on to look at a couple other studies that they have funded, and one by Gilman and al. In 2014 that they often will quote. It's often quoted that comparing 20 recreational cannabis users to 20 controls, by looking at their brain images, they concluded that there were differences in the shape and in the volume of the left nucleus accumbens and amygdala, a couple very important organs in the brain. No decrease in performance was studied, just the assertions of harm from the cannabis. They did admit at the end of the study that those subjects that were the pot smokers were also more likely to be drinking alcohol. So NIDA went into another study a year later, done by Weiland and al. In 2015. They did a larger and a better controlled study, now with 50 daily cannabis users and 50 non-users, as well as 29 adults and 29 adults, both users and 29 non-users. And they said that there was no statistically significant differences found in these and the other brain structures besides the amygdala and the nucleus accumbens. They said that when carefully controlling for alcohol use, no changes in the brain were found. This study is seldom quoted by the National Institute on Drug Abuse, showing basically no harm and really implicating alcohol and other drugs as a more risk. And then going back to look at things like short-term memory by a researcher by the name of Pope, short-term memory loss virtually disappears with brief periods of cannabis abstinence. And then in what's called a meta-analysis by Schreiner in 2012, they found no residual effects on cognitive performance in the cannabis smokers, again with just abstinence. There's a study by Hooper in 2014 on cognitive performance in adolescents, and they reported in 33 subjects abstaining from cannabis that there were no differences in IQ, attention, or memory. And in 2015, looking at the grade point averages, Meyer reported that lower grade point averages associated with cannabis usage lost statistical significance when controlling for alcohol and tobacco usage. And finally, I would just add another study in 2016 by Makaretz et al., and they said that IQ or performance differences in teens were not found associated with cannabis users compared to cigarette smoking or other confounds. So in doing harm studies, unless you're really controlling well for alcohol, tobacco, and other dangerous substances, you really can't see what's going on with the cannabis. And when you do control for that, it appears that cannabis is not harming the infants and children that are exposed to cannabis. Thanks for that question.
Oh, my pleasure. Well, thank you for the answer. The next question asks, what about impairment in driving when using cannabis?
Well, this is an excellent question because I do talk with employers, and they're concerned about their employees using cannabis on the job. We also are concerned about people driving. There are no simple road tests for impairment for cannabis, and there's a vast differences in the amount of cannabis that a person can have in their system, again, depending on tolerance that I spoke about earlier. So this is, I think the way to look at this is like any other drug. This is the only really reasonable way to look at it. We don't tell people that are using opioids or benzodiazepines or sleeping pills or any other kind of medicine, antipsychotics. We don't tell them that they can't drive the car. And if they are stopped and not performing well driving, they generally are not going to be taken from their car and have their car towed away. But this is being proposed now as a way to deal with doing roadside screening of cannabis users, simply to take their cars away. It's really illogical because there are studies that are showing that cannabis use in driving is really dependent on how comfortable you are with cannabis and how tolerant you have become to cannabis. So for a new user of cannabis, experimenting with cannabis, don't get behind the wheel. It's not the time to be driving. You will be spaced out and you might forget where you're going or just not be driving well. In general, the drivers that are using cannabis drive slower. They don't follow behind cars as closely. And generally speaking, they're safe drivers. There was an interesting study that came out of the National Highway Traffic Safety Association in 2010. It was a double-blind study, placebo-controlled, with 20 heavy cannabis users. And what they were doing was assessing the impact of alcohol and THC on these people's ability to critically track, meaning staying in their lane, a divided attention, and in a stop signal task where you've got to be able to brake at the proper time, and in cognition. And the conclusions of this NHTSA study was that alcohol significantly impairs those driving tasks, and THC did not affect those tasks' performances. So the present study generally confirms that heavy cannabis users develop tolerance to the impairing effects of THC on neurocognitive task performance. I just encourage people to be smart about cannabis use, and my patients will come and say to me, oh, if I use cannabis in the morning, I'm going to be sure to not drive for the next few hours. And I think that's generally safe and wise to do. Other people do use cannabis driving and are rather safe drivers. And if you look throughout the literature, you'll find that to be true as well. Thanks for that question.
Well, if there are no more questions, I would like to once again thank Dr. Hergenrather for his presentation. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I'd like to let everyone know that today's webcast will be available for on-demand viewing through June of 2018. You will receive an email from LabRoots letting you know when that webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thanks for joining us, and we hope to see you again here soon at LabRoots. Goodbye.